parents have a house in Rappahannock County.
Okay.
Yeah.
Nice. Just are you based in New York now? Is that right?
Sorry, you cut out for a second. Are you talking to me?
Where are you based?
Yeah, I'm in Brooklyn.
Brooklyn. And Ed, you're in DC?
Yeah, I'm in DC.
Terrific. All right. Well, we have people joining in. We had about a little over 700 people who who registered. So hi, everybody. As you join us, welcome. And I'd love to hear, just put in the chat where you're joining us in from. We have people all over the world and love to see where people are joining from. Great. And we'll just give a couple of minutes here to let people in and get things started. I see we have India and Germany, Mexico, Colorado, Englewood, Michigan, Montreal. This is great. I love having everybody from all over the world. So aloha from Hawaii. Aloha to you as well. Terrific. So we'll just wait about maybe 15, 30 more seconds, and then we'll get going here. All right. All right, well, welcome everybody. We're gonna go ahead and get started. And so welcome to the final science editing webinar from the Knight Science Journalism Fellowship at MIT. Today's topic is Fundamentals of Science from Funding to Publishing. My name is Joshua Hatch, and I'm your host today. I'm a former Knight Science Journalism Fellow and the editor of the Knight Science Journalism Science Editing Handbook, which you're going to hear more about in a moment. This is our 6th and final webinar of the year. If you missed our first 5, you can watch recordings of them at ksjhandbook.org. Just look for the menu that says webinars, and you can pick the webinar you want to rewatch. After the conclusion of today's webinar, you'll find this one there as well. All of these webinars are an extension of our online science editing handbook, which you can also find at, wait for it, ksjhandbook.org. I encourage you to visit where you can read the handbook online or download it as a PDF. And I'm also excited to tell you that we now have a Spanish translation available on the site as a PDF download, and we're looking to have other translations soon. We have some in the works. Both the handbook and this webinar series are made possible and made free to you thanks to support from the Kavli Foundation and the Howard Hughes Medical Institute's Department of Science Education. And they are an outgrowth of a several years long set of Kavli-supported in-person science editing workshops. So we're really pleased to be bringing this to a much bigger audience, a global audience. And I want to give a huge thank you to Kavli and HHMI, and of course, everybody at the KSJ Fellowship at MIT, which is hosting these webinars, and to SciCommX, which is helping to produce them. As with our other webinars, today's event will run 90 minutes. In a moment, I'm going to introduce our 3 terrific panelists, each of whom will talk for about 10 to 15 minutes, and then we'll move into a Q&A session. If you're participating on Zoom, you can use the QA button to send in questions, which I'll be monitoring, and then we'll pose them to the panelists. We'll also, we're recording this session and posting it on our website, so you can rewatch it there if you'd like. For those on Zoom, if you attend at least an hour, you will receive a certificate of attendance sometime in the next week or so. We're also sending around a survey to ask you what you thought and get your feedback. These surveys are really important to us, and they're brief, so I'd appreciate it if you could take the time to complete it and let us know what you thought. If you want to live tweet the webinar or ask questions via Twitter, you can use the hashtag KSJ Science Editing, and feel free to let others know they can follow along on the ksjhandbook.org/live site. Again, as a reminder, the webinar is being recorded and it will be posted on that website soon. All right, so now it's time to introduce our panelists. We're lucky to have 3 brilliant journalists with us today, and I'm very grateful that they agreed to participate, and I'm excited to hear what they have to say. So we're going to start today with Emily Anthes. Emily is a science journalist and author whose work has appeared in the New York Times, the New Yorker, The Atlantic, Wired, Nature, Slate, Business Week, and elsewhere. Her magazine features have won several awards, including the AAAS Kavli Science Journalism Award and the NASW Science and Society Journalism Award. She is also the author of The Great Indoors, The Surprising Science of How Buildings Shape Our Behavior, Health, and Happiness, and Frankenstein's Cat, Cuddling Up to Biotech's Brave New Beasts. Emily has a master's degree in science writing from MIT and a bachelor's in the history of science and medicine from Yale, where she also studied creative writing. She lives in Brooklyn, New York, and Emily will be speaking to us today about the publication process and how to read a scientific paper. Apoorva Mandavili is a reporter at the New York Times focusing on science and global health. She is the 2019 winner of the Victor Cohn Prize for Excellence in Medical Science Reporting and is one of our handbook's authors. She wrote the first chapter of the handbook. She is the founding editor-in-chief of Spectrum, an award-winning news site on autism science. She led the team there for 13 years and joined the Times in May 2020 after 2 years as a regular contributor. Apoorva has won numerous awards for her writing, and her work has been published in The Atlantic, Slate, The New Yorker Online, and in the anthology Best American Science and Nature Writing. Apoorva also co-founded Culture Dish, an organization dedicated to enhancing diversity in science journalism, and was the founding chair of the Diversity Committee for the National Association of Science Writers. Apoorva has a Master's of Arts degree from New York University, with a Master of Science degree in biochemistry from the University of Wisconsin at Madison, and she is fluent in 5 languages, which is 4 more than I am, I'm embarrassed to say. Apoorva will be speaking about science funding and false expertise. And the final leg of our webinar relay team is Ed Young. Ed is a science journalist who reports for The Atlantic and is based in Washington, D.C. For his coverage of the COVID-19 pandemic, he won the 2021 Pulitzer Prize in Explanatory Reporting, the 2020 Polk Award for Science Reporting, the Victor Cohn Prize for Medical Science Reporting, the Neil and Susan Sheehan Award for Investigative Journalism, the John P. McGovern Award for the American Medical Writers Association, and the AAAS Kavli Science Journalism Award for in-depth reporting. I Contain Multitudes is his first book. His first book looked at the amazing partnerships between animals and microbes and was a New York Times bestseller. He also has a Chatham Island black robin named after him. And today, Ed will talk about how the scientific process can go wrong using the pandemic and long COVID in particular as a case study. So again, I encourage you, please make ample use of the Q&A features, and we'll make sure to get to as many questions as we can. So now, without further ado, Emily, I'm going to turn it over to you.
Well, thanks so much for having me and to everyone for tuning in. It's amazing to see where you're all from. So I suspect that there's a pretty broad range of experience and expertise in this group. I am going to start with some basics, but then I hope to get into some nuances and tips that I think maybe will help everyone. So to start with some basics, the way that the vast majority of scientific results get communicated is through papers in peer-reviewed journals. These papers are essentially the currency that science produces, and if you are covering science or even part-time, you will spend a lot of time reading, vetting, thinking about, and analyzing scientific papers. And typically a single paper will describe an experiment or a set of experiments with some background on its rationale, as well as a description of the methods, the results, and an interpretation of the results. The traditional publishing process, though, moves in general at a very different speed than journalism does. So publishing a single paper can take months or more than months. It can take a year plus. And that's because there are a lot of different steps involved. So after conducting the research itself, which can be, in some cases, a lengthy process, scientists have to analyze the data, write it up, and then submit it to a journal. At some of the most selective journals, papers don't make it any farther than that. They just are sort of rejected outright. But if the editors think that a paper has promise, they will send it out to some other scientists for review. And these are the peers that you hear about in peer review. So typically, these reviewers are other scientists that have real expertise in at least one of the areas of the paper, whether that is the subject matter itself or perhaps it's the methods. So if someone submits a paper on, say, the neuroscience of altruism, maybe one reviewer will be an expert in that topic, and another reviewer maybe doesn't know much about altruism per se, but is an expert on how to analyze fMRI data, which this paper uses. And so when the process is working well, these reviewers will read the paper with a critical and analytical eye. Their job is to make sure that the methods and the experiments are sound, that the results add up, and that the conclusions the scientists draw are justified. After they do this, they can make a recommendation to the journal that it either accept or reject the paper outright, or what happens in many cases is they might recommend or request specific revisions or changes to the paper. So then the manuscript will go back to the scientific team, they will make those revisions, it will go back to the journal, and this could go back and forth for several rounds until the journal and the reviewers agree that the paper is up to snuff. Then the paper is formatted and proofed and scheduled for publication. Publication in the actual print journal can take a much longer time, additional months to a year. So often papers will be published online before they appear in a print issue. Once the paper is accepted, as publication approaches, journals might put out a press release about the paper or give journalists early access to it under what's known as an embargo. And what that means is that journalists can read the manuscript, they can start vetting it, sending it out to other experts to comment on, and they can report and, sorry about the sirens, they can report and write up their stories. And then as soon as the embargo lifts, their publications can, press the publish button and the paper is out at the same time the embargo lifts. So this whole model of embargoed science journalism and embargoes in general is controversial, and we can talk about some of that in the Q&A if you all have questions about it, but it is how a lot of science journalism works and is something that you will need to be familiar with. So that's sort of a brief description of what scientific papers literally are and how the process works. But papers are a lot more than, quote unquote, just a way to communicate results. They're also how scientists advance arguments, engage in debates, or sometimes even fights with other scientists. And they're how scientists advance their careers. So universities and other academic institutions typically make their hiring, promotion, tenure, and other decisions based in large part on a scientist's publication record. So how many papers they've published, how frequently they publish, what types of journals they're publishing in, is it the really prominent journals? And what that means is that there can be all sorts of perverse incentives when it comes to scientific publications. So some researchers might publish, very small incremental findings in a number of different papers to increase their publication count. Some researchers might massage or manipulate data in questionable ways. If they didn't experiment and didn't find any notable results, maybe they will slice and dice the data in different ways until they find something that they can report in a paper and get a paper out of. Journals have their own incentives, of course, to publish papers that get cited by a lot of other scientists or to publish papers that get a lot of news coverage. And so that in turn creates other pressure for scientists and researchers to do things like do studies and submit findings that might seem counterintuitive or seem likely to generate a lot of headlines. And what that can mean is that can distort the scientific literature. Because what it tends to mean is that, researchers who find that eating chocolate every day for breakfast reduces the risk of cancer are much more likely to get their paper published than, maybe the 12 research teams that found that that is not the case. So peer review and publication in a peer-reviewed journal is considered the gold standard for communicating scientific findings, and it is. But I say all this because it's important to remember that just because a paper or a result has been published in a peer-reviewed journal, even one of the most prominent, big name, most selective journals, does not mean that the paper is flawless or even solid or legitimate or whatever the finding it communicates is quote unquote correct. And partly that's because, scientists and reviewers are human and flawed. They make mistakes. They have their own biases and agendas. There are also researchers, certainly not all of them, but some who work in bad faith or manipulate their data. And there are even some who submit outright fraudulent results, photoshopped images, fabricated data. None of this is to say that the scientific literature is on the whole bad, but there are all sorts of flaws and limitations that are important to keep in mind even when you're reading peer-reviewed papers. And so that's why it's really important that when you're covering a paper, you don't just talk to or feature the researchers who did the work, but send the paper out to other scientists who are not affiliated with the research team to get their thoughts and to have them vet it. And that is especially important if you are covering findings that have not been published in a peer-reviewed journal. That's something that there is often legitimate reason to do. So findings that are presented at scientific conferences, for instance, might be newsworthy, but generally have not been peer-reviewed. And then, of course, during the pandemic, we have seen just an explosion of what are known as preprints. So as the name suggests, preprints are essentially, or at least they are intended to be, early versions of papers so that researchers can get their results out into the world. And the pandemic is a perfect example of when that might be important. Maybe we need to get the vaccine efficacy numbers out before, we wait 6 months for it to go through peer review. And in general, that is still the goal for researchers who post preprints online to eventually submit to and get published in peer-reviewed journals. But that is not always the case. So some preprints are very good and will go on to be accepted, to pass peer review with flying colors and appear in highly prominent journals. But some of it is sloppy or bad and will never be submitted to a journal. Some will get criticism when it's put up on a preprint server and will turn out to be so flawed that the researchers will sort of withdraw it or shelve it. So it's even more important if you or someone on your staff is covering a preprint to really make sure that you put it through some proper vetting and to get outside opinion on it. So with a little bit of time I have left, I want to talk briefly about how to read a scientific paper and how to get the most out of it. So you've probably heard some of the basics, but essentially you probably don't want to read a paper like a novel, that is, pick it up for the first time and read it straight from beginning to end. Usually it makes sense to, maybe start with the abstract, which gives you a quick overview, and then either go to the introduction, which will give you background on the experiment, or even to flip straight to the discussion or conclusion section. That will not only recap the findings, but we'll put them in context, talk about how they fit in with what else has been done in this field, what the implications are, and sort of help you have a big picture framework in mind that you can then use when you return to the rest of the paper and fill in some of the details. If you know how to read papers carefully, and if you do it enough, you'll also find that papers themselves have a lot of helpful clues that can guide your reporting. A few of sort of these tips that might be helpful starts right at the top with the list of authors. So, a paper might have 2 or 5 or 10 or 100 authors. And typically the way it works is the last author listed is the most senior author or scientist. Often the work was done in their lab. They were in charge of applying for and getting the grant. And they typically oversee the work at a high level and are really great sources for talking about the large big picture context, rationale for the study, how it fits in. But if you are looking for like really colorful, visceral details, like what it was like to conduct the experiment or what it smelled like when you added the red liquid to the blue liquid, you probably want to reach out to the first author who's listed. And that's because that's usually a junior scientist, often a graduate student or a research fellow, who actually did the sort of day in, day out work of the research and probably has the best familiarity with some of those nitty-gritty details. I guess while we're talking about details, one of my other favorite tips is although it can seem technical and dry, one of the best places to find color for your story is in the methods section. So I once wrote a feature story about the search for male birth control pill. And one of the researchers I was writing about had done some experiments on rabbits, giving them a drug that could stop sperm production. And it was a fairly straightforward scientific animal experiment paper, but sort of buried deep in the methods section was a note that the rabbits had been given this drug in a sugar syrup that was flavored like banana cream. And so that's just like a small detail. The story didn't rest on it, but it's the kind of fun little piece of color that can help really take your stories to the next level. And those details are often in either the methods section or also in what's known as the supplementary information, which is typically posted online and has a lot more nitty-gritty details about the methods of a particular study. Another thing you want to be sure to look for towards the end of the paper, typically before the citations, is what's typically called a competing interests section. It's often in fairly small print. It's easy to overlook, but good journals will ask authors to disclose potential conflicts of interest, like whether they're receiving funding from the pharmaceutical company whose drug they're studying. So it's really important to look for those. And if there are some concerning competing interests, to ask the researchers about them and to disclose them in your story. And then finally, the citations themselves can be a really helpful guide to other researchers or scientists that might be worth talking to, reaching out to get their thoughts on the paper. They're typically people who have done work in the same area. And one thing that I find particularly helpful is that often in the conclusions or discussion section of a paper, researchers might note that their results diverge from or conflict with the results of a previous experiment or study. So I always look up that study, both for my own information and for context about the diverging results or findings. But also the authors of those papers are often really good outside sources who might have a different lens on this particular paper or might be able to give you some crucial context that you're missing. I think that's all I've got. That was kind of a whirlwind tour. I'm obviously happy to talk in more detail about any of this stuff during the Q&A.
Thank you, Emily. I'm glad at the end there you mentioned other papers that are referenced because, correct me if I'm misstating this, but often scientific papers are part of an ongoing conversation or dialogue about some science. Is that not the case?
Yes, absolutely. And, I should have mentioned that, no paper or finding comes out of the blue. They're often very incremental. They build on work that's been done over years or decades and advance it forward in some way. And that's also where sort of what I alluded to about how papers are often how scientists wage debates and arguments. And so even though they seem sort of maybe buttoned up and technical, sometimes they're quite sort of knives out in terms of like trashing a prior theory or buttressing it.
Mm-hmm. Let me ask you another question that came out of the Q&A here. There's several, but one I wanted to get to was, I'm going to rephrase it, but there's a question here about what should be avoided in a pitch. And the reason I want to bring it here is because if I'm not mistaken, often a scientific paper will be sort of promoted by the university PR or something with a rather, or maybe an often rosy depiction of what the paper's findings are. How does that you speak a little bit to the need to read the paper to compare it to what the pitch or the PR folks are actually saying and how they do or don't line up?
Yeah, I mean, I would almost, this is a little bit extreme. So, I might not go quite this far, but I would almost say throw the press release away. I mean, like the press release might be how you found out about the paper. It might have landed in your inbox. But then you go read the paper and you report based on that. I mean, I know sometimes scientists I've talked to are themselves displeased with how their work gets communicated in a press release. So the paper and the scientists should be your sources. Maybe the press release was helpful for getting something on your radar, but it should not be the basis of your story or your reporting.
And I see Ed agrees in the chat there quite vociferously. So I had said that we would move to Porva next, but she's hasn't been able to join us yet for some reason. So we're going to actually, if it's okay with you, Ed, go ahead and switch it over to you next, if you don't mind. And your chat is a good segue to that. So Ed, if you're okay with it, we're going to move to you. Thank you,
Yeah,
Emily.
sure.
We'll come back with questions.
Great. Well, thanks for having me. I want to echo everything that Emily said. I think that's a fantastic intro to some of the systems that we as science journalists have to be aware of and to be mindful of. Emily talked a little bit about some of the dark sides of that process, like what happens when it goes wrong. And that's one thing that I want to talk about here. One of the most important things that I hope you take away from the session is that science is not just an abstract thing. It's not just a march of progress or just a series of objective truths. It is, in fact, an intensely human endeavor. It's done by people and it's subject to all the frailties and foibles that humans contain. And you can sort of see that in looking at how the scientific establishment reacted to COVID-19. So as of last December, when I wrote a piece on this, PubMed had archived more than 74,000 COVID-related papers, which is more than twice as many as there have been about diseases like polio, measles, cholera, and dengue. One journal got more COVID papers submitted to it than the total number of Ebola papers that have ever been published. So COVID drove an absolutely enormous pivot in the scientific world. A ton of scientists dropped whatever they were previously doing to work on this new thing. And some of that pivot was positive. A big pandemic threatens society. Of course, people want to help. They want to contribute in some way. As a consequence of that, a lot of folks who had previously been working on infectious diseases got new collaborators, they got new ideas. We collectively learned a ton about this new virus in a very short amount of time, far quicker than we had done for any previous new infectious disease. But there was a lot of negative, there were a lot of negative consequences too. Science funding is a zero-sum game. There's not an infinite amount of resources that are out there. And so when a new thing like COVID comes and grabs the world's attention, the funds that are shuttled into that get diverted from other things. Apuvo, who isn't with us yet, has written wonderfully, for example, about how research into tuberculosis, which is itself an enormous global health threat, was effectively paused during the early days of the pandemic. Many people in many fields have complained about how the COVID pivot diverted attention from their particular issues, many of which were, of course, also very high priority. We also saw a lot of what has been called epistemic trespassing, which is when people with expertise in one particular area barrel into areas that they know very little about, often with a huge amount of overconfidence and hubris. And that can make things a lot worse. It dramatically reduces the signal to noise ratio. In the case of COVID, there were a lot of studies that reinvented the wheel. I talked to a scientist named Lindsay Marr who works on aerosol transmission. The importance of aerosol transmission in the spread of respiratory illnesses became abundantly clear over the course of the last year, and many people are now taking it more seriously than they have done before. That's great, but a lot of this work just reinvents stuff that folks who've been in this field for a long time had already known and had already been doing. I asked Lindsay at the end of last year how many studies she had seen that had actually taught her something new about aerosol transmission of viruses. And after a very long pause, she said one. There are a lot of other studies that came out that were just misleading or shoddy. A lot of things that sent researchers down unproductive paths. The massive hype around things like hydroxychloroquine, a classic example, but there were loads of some of that occurred, I think, because a lot of folks were just stuck at home looking for ways to contribute. And because a lot of the same systems that have made science faster over the years, free data databases, the ability to publish work with preprints, the ability to publicize it on Twitter, all of that not only makes it easy to spread good science, but also makes it easy to do bad science if you don't actually have a sense of what your skill set is and how much there is to learn about a field to which you're new. I think a lot of the problems arose also because of the perverse academic incentives that Emily talked about. So academia essentially functions as a pyramid scheme. If you consider that every professor is going to train an average of 6 PhD students over her career, but only about 16% of those students are going to get tenure track positions. So the competition in academia is absolutely ferocious. And to make matters worse, the metric that most defines an academic success isn't publishing, immensely high quality, rigorous work that solves societal problems. It's publishing as many papers as possible in high profile journals. And that is easier if the work you are trying to publish is exciting, dramatic, novel, attention grabbing. Those incentives from academia don't push scientists towards rigorous, careful work that addresses massive problems like the pandemic. It actually pulls them towards practices like speed, cutting bits of research into small bite-sized pieces, cutting methodological corners, trying to do work as quickly as possible with smaller sample sizes, or perhaps doing what's known as p-hacking, where you alter your plan of analysis as you go along to get results that are going to be more publishable, more exciting. It pushes people towards hype, and talking to folks like us, to journalists, when doing that, it pushes them towards overselling the importance of their work or the quality of that work. And these are problems that exist at the very best of times. They've fueled what has been called the reproducibility crisis, where in a lot of different fields there is a growing awareness that a significant proportion of published work simply doesn't hold up, cannot be reproduced, is methodologically shoddy. But COVID intensified a lot of these incentives and a lot of these pressures. People who published work quickly and early on typically got a huge number of citations and a huge number of attention for it. The media, us, we intensified that too. We provide, by trying to meet the needs of our readers, this anxious population crying out for more information, we published a lot of work. We gave airtime to any expert who wanted it, and in a situation where any new paper, any new preprint can immediately draw international press coverage and hundreds of citations, it's no wonder that people are going to rush into this field, regardless of whether they are actually qualified or trained or any of the above. So science, of course, is meant to be self-correcting, and is sort of famous for that. So you might ask and wonder, why does it matter if the literature gets polluted by a lot of work that whether it is weak? Surely all of that comes out in the wash. And the answer is that it might do over time, but that process is also slow. And the same problematic structures in academia that we've already talked about mean that once bad work and bad ideas get into the literature and get into the public sphere, it's very hard to remove them. So I see this a lot on the topic of long COVID, which is something I've been reporting about for over a year now. In May and June of last year, when I first started thinking about this, most scientists and physicians had not heard of it. One person I asked, a very well-respected infectious disease person, when I described long COVID, said that's unusual. And even then, it wasn't unusual. Thousands of long haulers were already filling support groups, but there was this perception that this was very rare. The common wisdom was that the vast majority of COVID cases, aside from the ones that are severe enough to send people to hospitals, are mild, and that these mild cases resolve after 2 to 3 weeks. And this is kind of an astonishing statement to make for a disease that didn't exist before 2020. And yet this perception of COVID as a mostly mild disease, and that in which mild cases resolve quickly, very, very quickly became something quite close to dogma. And the consequence of that is that folks who didn't fall into that model, a lot of long haulers who had persistent symptoms early on in March or April of May of last year, were ignored. Like they didn't know what was going on with them. They were dismissed by a lot of the medical establishment who told them they didn't have COVID. After all, we know that COVID lasts for 2 weeks. And they often couldn't get tests, especially early on when testing was very rare and not available. A lot of long haulers just couldn't access it. And by the time they actually got their hands on tests, it was way too late. They were well beyond the acute phase of their illness, still struggling with persistent symptoms, but getting negative results that then screwed them over for months and more than a year to come. A lot of folks who have long COVID still don't have the surety of a positive PCR test. A lot of them have been excluded from studies and clinical trials that have since arisen about long COVID. A lot of them are struggling to access disability benefits. And almost perversely, the lack of a positive test is then used as a reason to doubt the entire existence of long COVID in the first place. This despite the fact that several groups have now looked at long haulers both with positive and negative tests and shown that the course of their symptoms is fundamentally the same. The main difference is that people with negative tests got their access to tests much later than those who had a positive result. That bias then skews the scientific literature itself. I've interviewed researchers who have tried, who have run long COVID clinics and who have tried to study this patient population. One of them, a guy called David Petrino, told me that he tried to publish a paper about the group of patients that he was looking after in his clinic. And one reviewer adamantly refused to let the paper through if it contained any results from patients who had negative PCR tests, which, as we've already discussed, is not it's not a solid contraindication. It's not a sign that people didn't have long COVID. But even though Petrino says that he was quite happy to analyze the data in his paper separately for positive and negative, and even though both of them presented with fundamentally the same symptoms, and there was no way that paper was getting through, and so he withdrew it. My point here is that these processes that we've talked about sometimes work to the betterment of our understanding, sometimes allow knowledge to progress in the way we expect it to, but sometimes it holds it back. Sometimes it allows a faulty understanding of the disease to calcify, and sometimes it allows shoddy work to pollute the literature for a long, long time. And I think that is really, really important for us to understand as science journalists, that when we look at the literature, when we talk to people, we have to really ask hard questions about all of the structures and the incentives and the processes that led to the kind of work that we're seeing in front of us. And science is often caricatured as purely empirical, objective pursuit. But actually, scientists' interpretation of the world around them is influenced by the data they collect, and that's influenced by the kinds of experiments they do, That's influenced by the kinds of questions they think to ask. And that's influenced by their identity, their values, the dogmas in their field, their imaginative capacity, and the environment in which they operate, the academic incentive, publishing pressures, media attention, funding opportunities. We need to be cognizant of all of that when we're doing our work if we're actually to provide our readers with something that's as close to truthful as possible. Thanks.
Thank you very much. Actually, Rudy asked a question I think follows up what you're discussing quite well, which is how to talk to people who basically what you're describing, at least in some form, which is that science is this evolutionary process, but it takes a while.
Sorry, Josh, you're really cracking up. I can't hear this question at all.
I'm sorry.
Okay, let's just give Josh a little minute. Did anyone else catch which particular question he was trying to read out? Oh, Julia, was it your question? All right, I'm going to read that out. So Julia Rudy's question is how to talk to people who say that science is always not sure in the results. In pandemics, it was especially strongly obvious for people when we had bad news about virus, very different from day to day. How should we deal with this as science communicators? How to persuade, should we persuade people? Okay. There is a lot to unpack here. Okay, so look, the way I try to approach this is science is almost necessarily an erratic, stumbling process, right? So it's not just a series of facts that build upon each other. It oscillates, like our understanding of the world shifts from one study to the next in often confusing and contradictory ways. And then gradually that process stumbles towards a state of reduced uncertainty that we might call the truth. But it's a slow process, and it is an erratic one. And I think that if we show every step along the way, the process becomes like stroboscopic, right? It becomes like dancing in a nightclub where you just see flickers of images around you. And I'm not sure that's of service to readers in the middle of a rapidly unfolding crisis like the pandemic. What we've tried to do at The Atlantic is to, rather than to do that, to try and do bigger and more broad pieces that look at a large swath of research at any given time, and that are a little bit more, I guess, proofed against the sort of vagaries, like this swaying process that research can take in a very chaotic moment. I think that provided just a more, I guess, like stable sense for our readers. I mean, look, the other part of this question was about persuading people, and I think that mirrors some stuff that other people have asked in the chat about how to convey the limitations and pitfalls of science without fueling the trend of science denial. I think fundamentally our job is to tell people what is happening, warts and all. I don't think we should be ever be afraid or reticent about being critical of science or talking about its limitations. And I think the model that we're sort of operating on there is wrong. The idea that you talk about the limitations of science and then you feel distrust. I think it's exactly the opposite way around. I think if you try and convey this idea of science as fully deserving of trust when it actually isn't, you diminish trust even further. Like, it's not our job as science journalists to make the case that science deserves trust. It's the job of science to deserve trust. And if it doesn't do that, then it's our job to say that.
I can chime in too.
And I apologize.
Oh, sorry. No,
no, Emily, go ahead, and then we'll. What's that? Why don't you hold it, and let's go ahead and toss to Apoorva,
Perfect.
and then we'll come back. And I apologize for my internet. I will be talking to my provider later today. But go ahead.
Thanks, Josh, and nice to be here. Nice to share this time with Ed and Emily. And I'm sorry I'm a little bit late to this. I am here to talk about funding, money, which is always a great topic. How is science funded and why does it matter? There are so many things about this pandemic that are different than how things normally operate, the pace and the fact that so much of the research is coming out in preprints, who qualifies as an expert or who thinks they qualify as an expert. But some things are the same, and money and the bias it brings is one of them. And it really matters because who funds the research can skew the findings, and not necessarily exactly what the results are, but how they are presented, what kind of analysis makes it into the paper or into the press release, and what gets left out. As Ed mentioned, science is self-correcting, but that takes time. And in a pandemic like this, when things are really fast, we don't really have the luxury to wait for science to correct itself. We kind of have to be even more vigilant than we normally are to pick apart what we're looking at and to see what biases there might be. Very broadly speaking, there are 3 sources of funding. I think this is pretty much true anywhere in the world, although there are some differences. The government, nonprofit organizations, and pharma companies or for-profit companies. Sometimes you'll hear that science is a very pure endeavor, that it's free of bias. And of course, that's nonsense because science is run by people. Companies are beholden to their stockholders, and everybody wants to see results. So money really does influence science. And it's very important when reading a study to look at the acknowledgements and see who funded the research. Another thing that's become incredibly important during this pandemic is to ask scientists if they have conflicts of interest. So, for example, anytime that I write about Moderna or J&J or Pfizer, and I talk to experts, it's become routine now to say, do you get money from Pfizer? Do you get money from Moderna? And you'd be surprised how many times the answer is yes. Sometimes they're unpaid advisors, but a lot of the time, they are actually paid consultants to these vaccine companies. And even the most pure of heart scientists would be uncomfortable speaking out against a company that gives them a ton of money every year. It's unconscious bias, even if it's not conscious bias. And even if it's not a real conflict of interest, even the perception of conflict can really sink your article and the credibility of what you're reporting on. So it's incredibly important to get to the bottom of the source of funding. In the US, the first one I mentioned, government, is really the main source of funding, and that's the National Institutes of Health. There are other agencies under the Health and Human Services auspices that fund research. The National Science Foundation funds biology and math and engineering, and the CDC funds some studies, but the bulk of biological research is funded by the NIH. And in other countries, there are equivalents. For example, in the UK, it's the Medical Research Council. In India, there's the Department of Biotechnology, but there is usually a government body that does much of the vetting of grants. The NIH is, of course, incredibly powerful, and whether directly or through collaborations, they also fund research in a lot of other places. But the funds that the NIH gives out actually go to universities, not to individuals. Often it'll go to a specific department, or it goes to the larger college if it's a smaller place. And the universities will keep a big proportion of that money as overhead costs. How much of that depends on the university? So often the researchers themselves will only see a small fraction of that money, and the rest comes from their department. So they need to keep their bosses at the universities happy. The government gives out money to the universities, and then the money is sort of divvied out for salaries, lab equipment, and for any work that their teams do. Sometimes there are specific grants that say you can hire one person to do this work. But for the most part, it's given out in these bulk packets. How much money is available for individual labs also depends on administrations and budget priorities. The NIH has done very, very well some years. They have gotten doubling of their budgets, and both Republican and Democrat administrations have been really good about prioritizing research. But there's no doubt that there are many, many more scientists who are applying to grants at any time than there is money. These days, the NIH funds about 1 in 10 grant applications. So the competition for who gets money is really fierce. And if you lose out on a couple of grants, you may have to end up closing your lab. The reason all of this is important is because the scientists who are in these labs feel enormous pressure to keep having their research look attractive enough for the NIH to fund, to have their results look good enough to continue to get grants, to continue to publish. You've heard a little bit about the publication process, but there is this idea of the minimum publishing unit. It's where universities, the researchers will publish the tiniest bit of new finding because they just want to seem like they have a lot of research funding, a lot of results that are coming out. They want to seem more impressive than they actually are. So if they can say, we published 25 papers this year, that sounds better than we published 2 good papers this year. So it's very important when looking at those papers to see they make it sound good, but is this really adding anything to the bigger picture, or is this an incremental finding? The same happens in grant applications. When researchers apply for a grant, they can make anything sound like it's important, and often do, just so that they can get a grant. So you might have somebody who works on fruit flies saying that their work is really relevant to cancer research. Things are really a stretch sometimes. And so that's also where it's important to ask the researchers exactly how is your paper relevant to this thing that you're saying it's relevant to. During the pandemic, if you look at a whole bunch of preprints, they all say things like, this paper has important implications for how the virus is contained. This paper has important public health guidance in the United States. And most of the time, the vast majority of the time, those papers really do not have any impact, and they're very tiny incremental findings. So the bottom line is really, when you talk to scientists, ask them what their funding source is and be skeptical. Apart from the government, there are these large nonprofit foundations like the Bill and Melinda Gates Foundation and the Global Fund that give out money to all over the world. And they have their own agendas. And they try to be very clear about what their goals are and what their metrics are. And then there are a lot of smaller nonprofits in very specific disease areas. Like, for example, there are a whole bunch of small organizations that fund autism research. But it's very important to look at those organizations and see what their agenda is. And there's a lot of information when you go to a nonprofit's website. You can see in the About Us section, or you can sometimes read between the lines and see that actually this is not a legitimate organization that is funding legitimate research. For example, in the autism world, it's very common for people who are anti-vax to say things like, we want to understand the role of the environment in autism, by which they really mean vaccines, because they believe that vaccines cause autism. So that kind of fine lens to see exactly what somebody's agenda might be becomes very important, especially when the stakes are as high as they are in this pandemic. With the exception of a few private nonprofits, most of these groups are really small, so it can be difficult to find out where their money comes from. Often they are also beholden to their funding sources or to their stockholders or whoever they have. And so it can be difficult to track the money, but there is information on websites whose job it is to look at their tax returns and include that information. Pharma companies are really the third big pile, and they're probably the most easy to identify as being biased because they're for profit. They have very clear guidelines about how they make their money, and it's their job really to convince you that their findings are good so that they can get more publicity and they can make more money. That is ultimately the bottom line. So anytime you are talking to a pharma company or pharma company rep or reading a pharma company press release, you have to remember all they care about is the profit. Pharma companies will often say that they are testing a really valuable drug or vaccine, and sometimes that's true. Sometimes something is the first in class and it is really making a huge difference to the world. But often what they're doing is testing something that is very similar to something already on the market. And they have their own tricks for making this seem better than it is. 75% of trials that the pharma companies run are run against a placebo. And you've probably been hearing all about how that's a really good thing. Every clinical trial should have a placebo arm. But that's really only true if it's a brand new drug or a brand new vaccine. When you have a bunch of products already on the market, what the company should really be doing is testing against those existing drugs or vaccines and say, does this new one add any benefit? Is it better or non-inferior, as they sometimes say? And the vast majority of the time, they won't be. Most cancer drugs, for example, really offer a very marginal benefit in survival. Sometimes it's on a matter of 3 weeks, 4 weeks. This is where the fine print becomes very important. And one crucial way of figuring out whether the trial result is believable is to look at what the FDA actually asked the companies to measure in that clinical trial. And that information is available in the original application that the companies make. They have to say, this is what we hope to find in this clinical trial. The reason that the FDA does that is because otherwise, when the results come out, the pharma companies can analyze the data any which way they want. If you're good at statistics, there are a million different ways to slice the data so that it looks best for you. And they can say that they saw this benefit or that benefit, and that they saw a benefit in this particular subgroup. There are a lot of ways in which pharma companies can and do try to make their products look better than they are. They also, when they write their papers, often the people who write them are, if not directly, indirectly funded by them. And so the results are framed in a very positive way. There are studies that show that the papers funded by pharma companies often have dramatically more positive conclusions than papers from academic organizations. They also make sure that unfavorable studies never get published. So those are just a few of the reasons that it is incredibly important to look at who is funding the research. This information is usually available at the end of a paper if the scientists are honest, but when you're doing an interview, it's also very important to just ask outright, where do you get your money from? I'll stop there.
Thank you, Apurva. And hopefully my audio is working better. I apologize for my dropped internet. I'm going to keep my video off just to reduce load, and hopefully it won't happen again. My apologies. Apurva, there was a question here from Abdullah Sani, who asks about the funding of science publishing and paying reviewers. Could you speak a little bit to what happens there or doesn't happen, and the impact that has on the process?
I'm not sure about paying reviewers, but they do ask authors to pay, and that can actually really contribute to the inequity between authors in certain researchers in certain countries and researchers in other countries. A lot of scientists in some resource poor nations can't afford the fees that these journals demand. Some of the for-profit journals have somewhat legitimate reasons for asking for that money because they have professional editors and they say that they do a better job with, for example, copy editing, or they send out to good publishers. But the fact of the matter is that ultimately that really makes the platform very uneven, and institutions that will subsidize publication fees for their researchers will do better than a researcher in a developing country. So that is a huge problem in publishing. There are some platforms now that are open access, and they've really tried to make the whole process more democratic, but so far, really, none of the purely open access journals have become competitive enough with the really big names like Science or Nature or Cell. And when scientists are being evaluated for tenure or for any kind of promotion, or even for grants, when they apply for renewal of grants, what the reviewers in the universities or in the NIH look at is publication records. And if you have a lot of papers in Science and Cell and Nature, you look great. And if you don't, it's really difficult. And so that is another way in which research becomes very siloed into these high profile, high impact journals, and then everything else.
Perfect, thank you. And Emily, I know you were answering the question earlier before I threw it to Purva, so I want to give you a chance to finish that answer, and I apologize for cutting you off before.
Oh, not a problem. Yeah, Ed was just taking a question that had come up in the Q&A, and part of the question said something like, how do you balance communicating like the uncertainty of science and the limitations without like giving fuel to science denialists? And I was just, Ed had like a lovely answer. I was just going to add that I think that's a bit of a false dichotomy, and that you sometimes see, and I think I've seen a lot in this pandemic, this sort of paternalism that says, oh, like the public can't understand nuance or complexity, so we're not going to bother with it. I think the most dramatic example, I know lots of people in this webinar are not in the US, but those of you who are may remember, you know, the CDC saying very early on, masks don't work, masks are useless, and sort of very conclusively. And that was because of real concerns about supply. I think that, you know, people would go out and hoard masks and there wouldn't be masks left for healthcare workers. Well, once it turned out that like, oh, maybe the virus does spread through aerosols and masks would be helpful, that is now a much harder message to convey. And what has happened is people say like, see, scientists don't know anything, experts don't know anything, they were wrong about masks. And so I think really like, if you want to build trust in science and the scientific process, like you do that by being open and honest about the limitations and uncertainties, and that is sort of the only way you could build that kind of trust.
Yeah, that's a great example. So thank you for that. I'm going to pull up some of the questions that have been asked here and put them to you, Emily, and to you, Purva. So there was somebody here who asked anonymously, for the first time during the pandemic, I realized that people in general may not understand the phrase peer reviewed. When I saw social media comments like, I don't want something to be reviewed by peers, I want it to be reviewed by experts. Do you end up writing for people who already understand these concepts? And if not, how much explaining do you need to do for the general public who may not understand these terms?
I can answer that. I mean, at the times, what we try to do a lot of the time is say, if something is published in a scientific journal, we don't go into a lot of detail and say it's been peer reviewed. But when we cover a preprint, we try to say very high up, maybe in the second or third paragraph, that the results have not been vetted by other experts. We try to say things like that rather than use the words peer review, so that it becomes clear what we're talking about is this hasn't passed a certain bar where other scientists have seen it and said it's good. I think that's a easier way to do it than to throw terms at people that they may or may not know. It's like everything else in science journalism, the less jargon, the better.
Yeah, agreed. So let me, there was a question here, again, another anonymous. How do you choose what papers to actually cover? I've been told by editors that a paper I pitch only moves the conversation incrementally and hence is not worth covering. And at the same time, another person asked a similar question, although of kind of the opposite example, which is the reporter sort of recognizes this is not a particularly important or it's just an incremental paper. If your editor is for whatever reason hot to trot on it. Can you speak a little bit about what you choose to cover and how you have those conversations with your editors?
I mean, I think part of it depends on what your specific role and beat and goal is. Like some things during the pandemic are sort of self-evidently important, like the vaccine efficacy numbers, like regardless of what they were going to be, they were going to be covered. Sometimes though, like in non-pandemic times, and if those times return, we're also we're journalists and storytellers. So sometimes a paper is just really interesting, or like there's an interesting backstory there and it makes a colorful, great story, even though it might be about something that's sort of obscure. So it depends a bit about on what the circumstances are. In terms of having conversations with editors, I mean, a good editor, and I know we don't always have that luxury, but if you tell a good editor that you have vetted a paper, you have sent it to XYZ experts, and they all have concerns about it, they will listen to you and respect the reporting that you've done. I mean, it's hard, you know, some editors are unreasonable for all sorts of reasons, but I mean, I think if you can show them that you did the work, like I reached out to people that know more about this than either of us do, and here's what they said. I guess that would be my suggestion.
I think that also works for papers that you do want to cover. You know, it's useful to have the names of several experts in your back pocket who you can email and you have an understanding with, that you're sending them the paper, not because you're going to cover it, not because you necessarily want them to comment, but just to have a quick look and say, this is crap, or this is actually worth covering. Obviously you don't need to do that for every paper. As Emily said, some are really obviously good and some are obviously bad, but there's a whole middle ground where it's hard to know exactly how big a deal something is if you're not intimately familiar with that field. The other thing that I think also becomes relevant there is this whole idea of a preprint and peer review. As we were talking about before, people have a limited idea of what peer review means. And so one problem that I've noticed on social media now is that some people have learned now what peer review means. And so they've taken that and applied it across the board to everything. And any preprint that you even think about covering, they say, well, it's not been peer reviewed, so it's not right. That's actually not how it works either. It's not that if it's not been peer reviewed, it's wrong. It just means it hasn't gone through the full vetting process. Peer review isn't perfect either. Lots of terrible papers make it through peer review, and lots of good papers get rejected and stay in a journal's pipeline forever. What I think is more useful is to send whatever paper it is, whether it's a published paper or whether it's a preprint, to a bunch of people who are real experts in that field and get their take on whether it's good. In a way, when you cover a preprint, you can almost do your own peer review. And of course, it's not as long and drawn out as a real peer review would be, but it still gets somewhere there. So it sort of shows the danger of people learning halfway what a concept is and then overusing it.
Isn't that true as well, that there are certain categories of papers that actually never get published or never get pushed forward? I'm thinking of things like replication studies or papers that maybe, or I should say research that maybe has negative results. And so maybe the incentive to publish isn't there. And yet those are still important findings. Can you speak a little bit to how that fits into this larger world that we're talking about?
Yeah, I mean, that's sometimes called the file drawer problem, where like if you look into something that might be interesting and you find no effect or something doesn't work, scientists just like stick it in the file drawer and don't publish it. And that's a bit about what I was alluding to when I say that journals had their own incentives. So like journals also want to publish papers that make the news or get cited or make waves. And so either, it's sort of this vicious cycle where like they are less likely, some of them, to accept papers that like don't find anything interesting. And then scientists become less likely to even bother writing up those findings and submitting those kinds of papers, because like, what's the point if it's not gonna get published somewhere? And so that's where you have this literature that gets distorted towards like these one-off counterintuitive or sort of sexy, notable findings where you're not seeing all of the like null effects that are being found.
Terrific. And sort of also on this sort of theme, Julia Beverly asks, what sites monitor retractions and how helpful are those for reporters and editors?
There is a site that actually tracks retractions. It's called Retraction Watch. It's easy to find. And it's run by Ivan Oransky, who's a prominent science journalist and who actually took my old job when I left. But anyway, it's a really great resource. It's a great resource to see which papers are being retracted, but they also do some misconduct reporting. So it's a really good site in general to keep an eye on what is not panning out in research and which people not to trust.
And yeah, Ed talked about, some of the problems with thinking that science is self-correcting. And like one piece of that is like, even when papers get retracted, like those retractions tend, if they're prominent, get way less coverage than the original paper did. And so it's like sort of hard to get the genie back in the bottle, as it were.
Very good. Kim Rogers asks, do you have tips on how to find experts who are not the authors or co-authors of a paper who are in a position to comment on a paper? I'll just put insert a little promo for ourselves, which is the handbook actually has a chapter on this written by Melinda Winter Moyer. You can find it on the website. But Apoorva or Emily, is there anything that you would like to say here about that?
Yeah, I mean, I find experts all the time in PubMed. You know, I look for people who have published similar papers on related topics. I usually don't just go by that one paper or two papers that I might find. I look for somebody who's published a bunch of stuff, and then I vet them, see, you know, what kind of other things they've done, you know, Google them, basically actually make sure that they're legitimate and good. And these days, to make sure that I interview enough women, I've also taken to Googling related articles and then the words "she said" in quotes so that it pops up a female expert that I can interview. That seems to be the only way sometimes that you can actually find the women who are experts in a particular field, because they're a lot shyer about demanding credit for the things they do.
That's a great tip. There's a question here from Angela Anandapa. As a scientist working in industry, one of the biggest challenges we've seen is the propagation of bad science in social media or by some quote unquote journalists that is not countered soon enough through other means. Can you comment on how to tackle that? I mean, I think basically asking about how to counter, basically bad news.
Yeah, I mean, it's hard, and it's something that we talk about often when you see shoddy studies getting attention on social media or somewhere. I think there's a fine line between giving something more oxygen often is not what you want to do. And so it's like, is it becoming widespread enough? Is this misinformation becoming widespread enough that it's worth countering it? I've seen one or two papers that were really poorly done and attracting a lot of criticism on Twitter. But when I looked into it, it was really just a few experts in the field that were critical, and rightly so, but decided that, putting something in the New York Times about it was likely to bring more attention to this shoddy work than it deserved. And so I think you have to think about that balance. There are times when it is worth it, when some bad science has gotten enough traction to be causing harm or spreading rapidly. I don't know that's a very satisfying answer, but it's a hard problem.
We actually have a misinformation desk at the Times whose job it is to track these kinds of things and then address them very directly. And they actually, what Emily said is one of the things that they really emphasize is, they keep an eye on something that is trending, some piece of bad information that's making the rounds, and they don't decide to cover it until it has become big enough that it's really worth countering, because if you do it too soon, you actually end up amplifying something that maybe should just be quiet. But if you do it too late, then that particular myth may take root, because often these are very simple, easy concepts, the myths, whereas science is messy and complicated and doesn't take root as easily. So it's important to do that. And one other thing that I learned from them that I think is cool and easy to do is to address the myth head on in the headline, because that's what gets searched by Google or whatever else. So their headlines will be things like, "No, the Pfizer vaccine does not affect women's fertility." They don't really mess about with subtle headlines.
I should just point out there are other people who are offering some websites and other tips in the chat. So for those of you who are asking some of these questions, please, and following along, please take a look at the chat for some of those. So another question here that in some respects is sort of the animating question of this entire webinar series and the handbook itself comes from John Paul Muro. How can editors help reporters who are not covering science-related issues, especially during this time of the pandemic, since we've seen almost 60 to 70% of newsroom reporters who are covering other beats having to turn to health, and I would argue other science-related topics like climate change. That's my own parenthetical, being the majority of issues revolving around health and COVID-19 globally. So, basically, what can editors do, and from your perspective as science journalists, to help those who are not deep into these topic areas do a better job. And of course, one answer would be refer to the handbook. But aside from my own plug there, what do you two have to say?
I can say what I used to do as an editor, although of course I was not at a place where people didn't write about science, but I think the more complicated the topic generally, and what we can sort of say is that even if science is not necessarily always more complicated than something like politics, it is a more niche discipline. And so it does take some expertise to know what you don't know. So if you are an editor who's commissioned an article on science, I would say, first of all, give your reporter a little bit more time to dig deep. Often an article on science is not as simple as this is what the study said, end of story. If you want them to do a reasonable job of pointing out the flaws and talking to enough people, then spend some time on it and make sure, in a way that's not necessarily the case for other disciplines. In science, you really have to have independent experts comment on something for it to be credible. So make sure that you have your reporters call multiple sources, not just the people who did the research. And then there are all these websites, and exactly, look in the handbook, and there are tons of websites that your reporters can go to, things like PubMed. And, if you can help them make connections to science reporters at other papers or magazines who do have expertise, the National Association of Science Writers is a very good resource. There are tons of people there who are friendly and helpful, and they also have a lot of toolkits to help people. And then there's a website called the Open Notebook, which has a ton of articles also on how to cover science, all the different sort of parts of science, how to write about science when you've never written about it before, everything else like that. So there are a lot of resources that the reporters can get to.
Perfect.
Yeah, Apoorva said this, and number one is more time, which is a hard thing to do in this news environment. But listen to your reporters if they say they need another day to get more sources. Some of the worst and most viral misinformation or stories has been stuff that was rushed out. And that's something you can do as an editor, is more time.
Terrific. There's a question here from Alia Utuova, and I hope I pronounced that correctly, and apologies if I haven't, about fact checking. And actually, I think there were a couple other questions about fact checking as well. I will point out that one of the sister projects of the KSJ Science Editing Handbook is actually a KSJ fact checking guide. So you can go and if you go to the handbook, you'll see a link to the fact checking guide. But the question specifically that Leah asks is more about sort of self fact checking. Do you have a process that you use or tips that you can offer on how to do sort of fact checking for yourself?
I mean, I do a ton of this. I don't know how much of this is, I feel like this is one of those writing processes that is very idiosyncratic. But if I have the time, and I'm working on a story right now, and I'm nervous about, I will copy the entire edited story into a new file and go through bit by bit and either gray out or delete or, somehow mark facts off as I check them, just going back through my interview notes, back through papers. I almost always find something, even if it's just a date or it's 18 instead of 17. So that's how I do it. But I think your mileage may vary.
Great. And here's this is maybe a question that sort of takes a step back a little bit. Some of the attendees here, we have a variety of attendees, some earlier career, some later career. And one of the questions here from a Jamie Zurchin, again, apologies if I didn't get that right. How do you structure your workday? Can you speak to that a little bit?
Apoorva?
I think it's another thing that's very idiosyncratic. I don't know if anybody wants to live their life like I do. I don't think it's very healthy. How do I structure my day? I actually look online the second I wake up because I'm like that. And I want to know what's been happening in the world. Is there anything that, did my editor contact me? Did all hell break loose? Did I get a really important fact wrong the previous day, whatever? So I might look very quickly at my email, look very quickly at Twitter, and at our company Slack and things like that. And then, I take my time. I have a lot of processes to make sure that I'm not distracted while I am working. So for example, I get a bazillion emails during the day, and I'm an inbox zero person. So if I see my emails coming in, I get very antsy. So what I do actually is take them all and dump them into a folder called unread news, which I know is not that creative, but that's what it is. And at the end of the day, I'll go and look at all the unread news. And obviously I only do that with anything that I think is non-urgent, pitches, emails from scientists who are like, oh, I want to talk to you about my work, things like that are not super urgent, go in there. And these days I actually write in our content management system. This is something that I only started doing at the Times, and it is a time saver because I'm not then transferring things over. So I would say on any given day, the bulk of the time goes to either writing a story or reporting a story. But even on days when I'm on deadline, I make sure to make at least 1 or 2 forays into other stories I'm working on. Like I might just set up a couple of phone calls. I might just read one paper. I might just do a tiny bit of progress, but I try to make progress on the other stories, because otherwise I only end up doing that one story and that's it. I never get to the other stuff. So I think that's one of the things I've learned over the years to keep myself productive. And inbox zero works for me, but different things work for different people.
Emily, do you want to take a shot at how you structure your day?
Yeah, I mean, so I do it slightly differently than Apoorva, but I also basically batch my emails and like sort of keep an eye on it during the day. If anything's urgent, I respond, but otherwise at the end of the day, like, where the workday is like not quite over, maybe it is, but your brain's fried and you definitely can't write anymore. Like that's when I respond to emails. I mean, I guess it's so variable, but I think it just comes down to like paying attention to and knowing your schedule. So like since I started doing Breaking Pandemic stuff, I found that, if I want to schedule any other interviews for like longer term stories, it's best for me to do it in the morning, like in the first hour or two, because I'm much less likely to be derailed by like anything breaking that comes up. So when I'm scheduling, interviews for features, I try to do them at like, 9 a.m. But that's specific to the job I have right now. So I guess I would encourage people to pay attention to their own daily rhythms, maybe for a week or two, really closely. And then you can figure out sort of when it makes sense to slot certain things in. I was a freelancer for 15 years before this, and like my schedule is very different.
We could probably do a whole webinar on email management and
right.
Apoorva. I think I'm an Inbox Zero fan, and it looks like a couple others are as well, but I know it's not for everybody.
It's also been very hard in the pandemic. It's like Inbox Zero has become a whole new level during this pandemic. It's very hard to do.
Aspirational, at least. Mm. We're sort of getting to the end here, just a few more minutes, but there's a question here from Alucin Rain Wilson, who basically is asking a question, I'll just paraphrase, about how do you get scientists who are very busy to take time out of their day to talk to you, especially, and I know that in our webinar on sourcing this came up, maybe they don't think of themselves as being worthy of being quoted and being a source for your story. Do you have tips to convince them otherwise, I guess?
I mean, these days, it's basically you write a New York Times reporter and they will respond, but I have not always worked at the New York Times. So, and I know what it's like to try and reach people when you're not at a big name. One thing I have done is give them enough time. So, I'm not always working on deadline on stories, and I give them as much time as possible. And I put in the email, I'm very clear about, or sometimes even in the subject line, how much time I need. So I'll say, I will, this shouldn't take more than 10 minutes of your time, or more than 15 minutes of your time, or whatever. So that the ask is very limited and they know that I'm not going to just keep them on the phone for hours. So I'm clear about that. I'm clear about what I'm writing about. I have a whole bunch of email templates, and I can just, pull one in that says I'm working on X, and then I fill in the X. And then I also often have, if it's somebody that doesn't know me at all, I will have like a link to a couple of stories that are similar so that they can see what kind of work I've done and that I'm reliable and won't like completely mess up the science. And then once you start to write on a particular topic, or once you start to get to know sources, I think it snowballs pretty quickly too, because people get to know who you are and people get to know that you're reliable, and they are more likely to write to you. But having that subject line be very clear, like, time sensitive interview request for X, is super important.
Just quickly, I think that's another side benefit of really working to diversify your sources, because if you're going to head of Harvard Institute or Nobel laureate, they have a million interview requests and demands for their time. And so by looking for people who are not in the media all the time, will both have more time for you and be more interested and willing to speak.
Terrific. I think we'll probably have time for just one or two last questions here. And I'm kind of going to bring it back to where we started. And there was a couple questions here about open access journals. And one of them is simply, why do they not compare to the big name journals in the same way? What's holding them back? And how should science journalists weigh these journals against the bigger ones while reporting? So maybe that's something that, Emily, you could touch on.
I mean, I don't know deep in the economics of all the open access journals, but my impression is money and tradition are the main obstacles. The traditional journals make a lot of money from subscriptions, and they also have, the big ones have been around for a long time. So they have these names and reputations already established. In terms of vetting, I don't tend to pay a lot of attention to open access versus non-open access, per se. There are some open access journals that are quite good and some that are not, and the same with non-open journals. It's more about the journal itself, and what kind of reputation they have, what other types of work I've seen in there.
I think it's important to look beyond the brand name, because science and Nature have been around for a long time, but I used to work at Nature, so I can say this: profit is still the biggest driver. So those companies have just proliferated more and more and more journals as time has gone on, and they're not all the same quality. Nature might still be excellent, but Nature has all these other new branches now. Science has a new one called Science Reports, which is not very good. The way that a lot of these companies work too is that they have a watering down system. You might apply to Nature. If Nature doesn't want it, it'll punt it down to a sister journal that's lesser quality, and lesser quality, or I shouldn't say lesser quality necessarily, but lower impact factor, and it'll keep going down the chain. So it's kind of helpful to know what that hierarchy is, and you can know that hierarchy by asking other scientists when you talk to them. And it's also a mistake, I think, to think that Nature, Science, Cell are only the big journals to look at, because they are very broad. They have a very broad span of topics, and they may only publish the things that they think are of broad interest. But often the really interesting stuff happens in journals that are limited to that field. And if you have a beat, it's really good to know what those journals are in your beat. If you're reporting on something new, ask the first expert you talk to. What are the journals you read regularly? What do you get a subscription to? Because sometimes for people in that field, publishing in that smaller niche journal has more value and higher impact than publishing in Nature or Science.
Well, that's a terrific tip, and I think a great place to end the webinar. We're at time. Thank you so much, Emily, Apoorva. Thank you to Ed, and thanks to everybody who attended today. This is made possible because of all of you. It's also made possible thanks to the Kavli Foundation and the Howard Hughes Medical Institute's Department of Science Education. Again, as a reminder, this webinar was recorded, and we will post it to ksjhandbook.org very soon. For those of you who are going to be looking for your certificate of attendance, that'll come sometime in the next week, as will a survey, which we really would appreciate you taking just a few minutes to fill out. If you haven't already, I highly encourage you to check out the other webinars, all of which we recorded and posted on the site, as well as the handbook itself. So on behalf of KSJ, thank you, Apoorva, thank you, Emily, thank you, Ed, thank you everyone who attended. I hope you stay healthy and have a great day. Thanks so much. Take care.
Thanks for coming.
Thank you.
Bye-bye.